Navigating Biochemistry Research, a guide for undergraduates. So just like the moderator said, just like the moderator earlier said, building the researchers is focused on helping Nigerian undergraduates in the biomedical, biological, and life sciences field. Okay, so tonight's webinar will be focused majorly on biochemistry research. And this is to give you a basic knowledge, additional knowledge rather, that you need as an undergraduate in biochemistry. Okay, so we're working through it, and I would like to jot down your questions as we proceed. And you can jot down questions from this particular webinar and probably questions that you've had before on how to navigate your undergraduate research. So let's start. Yes, so what is research? From here, I have it here that research is a systematic process of investigation and inquiry aimed at advancing knowledge and understanding in various fields and disciplines. Now, I'm going to break this down. From this definition, we have advancing, we have knowledge, and we have understanding. So we know research is like a common word, right? And people just talk about research, and probably what comes to your mind when you hear research is, oh, lab work, or research. That means you're doing experiments and all of that. But before we get to lab work, before we get to experiments, or before we get to sacrificing rats and all of that, research in itself simply means you trying to find out something, you trying to gain more knowledge about something, you trying to increase your understanding of a particular thing. It could be anything. You could actually research, you could make research on Instagram, for example. I'm trying to like explain to you that when we say research, it's not only limited to science and it's not only limited to lab, right? It simply means you are researching, you are searching, right? You are searching to get to know more. So that's research generally. And research can be on different fields. You could just be at home and you hear a word, or you just see something on the news and you want to get to know more about it, and you pick up your phone and you browse it. That's you doing research. Okay, so that's research basically. Now, research is essential in various fields, including science, business, social science, like I've explained. Even as someone that is very social, or even someone who is a content creator to start with, you want to get to know more things about content creation as a field. You can carry out research, you can browse. All right, you browsing, that action in itself is research. But now let's talk first about types of research. Number one, basic research. Basic research aims to simply increase your knowledge and understanding in any specific field. So when you just pick up your phone and you just, oh, I've been hearing this word. I've been hearing the word education. Yeah, I've been hearing the word education. What is education? Let me just find out. And then you pick up your phone and you just check Google. Not only your phone, mind you, you can also get books from the library and you're searching for education. Or let's even come to the primary. You pick up a dictionary and you open to search for education, and you're like, oh, what's the meaning of the word education? That is basic research. You're not going deep into it. You just want to have preliminary knowledge, right, about a particular thing. That's what basic research means. Now, when we talk about applied research is focused on solving practical problems or addressing specific needs. It aims to apply existing knowledge to real world situations. Now, this is pointing more to what we are talking about. It's pointing more to biochemistry research, because whatever we do in the lab, whatever we do as a biochemist, is to provide a solution, to provide real world solution, right? That is we applying research. So applied research is more or less like basic research with a mindset of gaining in-depth knowledge to be applied in the real world. So that is applied research. Applied research is beyond you just checking up the meaning of a word. It's beyond you just reading up some books and just say, oh, I know it now, I have it in my brain. No. Applied research goes further. You need to highlight, you need to document, you need to then see how that you've gotten from your research can be used in real time, how it can provide solution, how it affects our lives, our daily life, right? Now we have quantitative research, and then we have qualitative research. I'm picking it together because they are like two contrasting words, right? Now, quantitative research uses numerical data and statistical analysis to quantify relationships of different variables, patterns, and trend. Now, there is a need for me to mention here that as a biochemistry student, you don't just say, oh, I'm doing applied research. No, I need to also mention that these types of research is just to give us a picture, right? But all of these things come together to form what we do as biochemistry students. For example, if you are given a title or your lecturer tells you, okay, or yeah, or after you have been allocated to project supervisors, and then your supervisor says, okay, I'm actually working on lipids. What do you know about lipids? And you say, oh, I really don't know much about lipids. And then your supervisor goes ahead to say, oh, go and study about lipids. That's basic, right? That's basic research. You just want to know it. Then when we will not get to a point where we are going to get certain values. That is lab work now. We are going to get certain values. These values is the quantitative part of it.
Together.
This numerical data, statistical analysis that is a part of biochemistry research is quantitative. It's just like the word quantity, right? When you hear quantity, let what comes to your mind be numbers. Then we go further to talk about qualitative. Qualitative research is a kind of research that derives non-numerical and uses non-numerical data. This has to do with observational studies. It has to do with text, images, your understanding, your general subjective understanding. It does not have to do with numbers. Now, as a biochemistry student, you do not only work with statistics, you do not only work with numbers here. We also do qualitative research. I'll use my undergraduate study as an example. So we wanted to investigate, or we investigated rather, we investigated the edibility of racrepitan seed oil. And then after sacrificing the rats and carrying out biochemical assays, we got the values, the numerical values, right? That was the quantitative research. And then there was a need for us to also document the reactions, how that when these rats fed on the oil, what was how was their reaction to it? Were there reactions on their skin? All of those observational factors. And then our lecturer was asking us, oh, you people didn't take note of that? Were their air dropping? How about their transit time? Did they like defecate more? All of these things is still a part of the research. So, but in general, there are certain research that only does qualitative. That is, for example, you just want to investigate the idea of people, or the opinion of people on a particular matter. And that's just what you need. You might want to investigate the opinion of people on different lecturers. That's just like qualitative research alone. But as a biochemistry student, I'm saying it now that you will most likely do a work that would include everything. We have talked about basic, we have talked about applying it to real world issues, we have talked about quantitative, that's the values you get from your biochemical assays. We've talked about qualitative, you have to observe, you have to see how your rats are doing. I'm not saying that all biochemistry research will have to do with rats though, but majority of it. Then we have few others. We have experimental research. This involves manipulation of variables and measurement of outcomes. It aims to establish cause and effect relationship. Basically, I'll just say what you do in the lab when you perform experiments. Okay, if I add this to this, what happens? So that's just it for our field, right? Is the lab work part of it is the experimental research. Then we have descriptive research. This aims to describe and summarize existing phenomena, right? It provides accurate details of a particular situation or population. That is just descriptive. It does not necessarily have to be quantitative, but sometimes you might carry out a quantitative research that you then use for a descriptive research. That is, after getting your values, you then use the values to describe that phenomenon. That's why I said all of these types of research are actually intertwined. Then we have survey research. Survey research, a good example is your questionnaires. All right. It is used to collect data through self-report measures such as questionnaires or interviews. It aims to describe attitudes, behaviors, or characteristics of a population. In most cases, survey research falls under qualitative. Okay, but when we are working with structured interviews, structured interviews can also be linked to quantitative research. But it's all intertwined, right? Then we have historical research. This one just wants to go back in time and fetch what has happened before, want to talk about past events and all of that. Now, there are still other types of research. You can do more studies on it, but it's a good thing that I've been able to establish that for your biochemistry research or biochemical research as undergraduates, you have to have the knowledge of basic research, you have to have knowledge of qualitative research, you have to understand quantitative research, and then you have to understand that you are doing an applied research, because whatever we find out will be related to the real world, will be related to our daily living. Okay. So, like I noted here, it is worthy of note that many studies may employ a combination of research approaches to achieve their research objectives. All right, let's proceed. So now, what is biochemical research? What is biochemical research? We have established that research spreads across different fields, okay? But when we are talking about biochemical research, this is our own thing. What does it mean? In simple terms, biochemical research involves the study of the chemical processes that occur within living organisms, including structure, function, and interaction of biomolecules such as proteins, carbohydrates, lipids, and nucleic acids. Now, as a biochemistry student, there are all of these things that are highlighted here are things that you should be familiar with. Of course, when you hear biochemistry, you already know it's talking about the chemistry of the body, chemistry of living organisms. Okay, so when we are now doing biochemical research, what we want to do is to investigate this chemistry of this living of our living system, of our body. Let me just put it in layman time. Chemistry of our body. You want to investigate the chemical reactions taking place inside your body. Now, what are the reactants? What are those chemicals? They are the biomolecules. In simple terms, biomolecules. And your biomolecules are your proteins, carbohydrates, lipids, and nucleic acids. Now, in biochemical research, you want to know, okay, if I introduce this particular chemical, or if I introduce this particular food, of course, even food is a chemical when you break it down. So, simply put, if I introduce this chemical to a biological system, to a living organism, what happens to the pre-existing state of that living organism? That is to say, okay, for every normal human being, this is how this is what the lipid level should look like. This is how the protein should look like. This is what nucleic acid should look like. Now, if I decide to introduce an external factor that is a chemical, now what then happens to how does this scatter or improve the pre-existing state of it? How does this scatter or improve the protein, the carbohydrate, and nucleic acid how they were before? How does it alter it? This is what we are doing. We're talking about biochemical research. So that's just like the simplest. That's just like the simple explanation for it. It aims to elucidate the molecular mechanisms underlying cellular processes such as metabolism, signaling, gene expression. All of these things are biochemical terms. But of course, when we are talking about gene, talking about metabolism, talking about signaling, everything that brings about all of these things are still biomolecules and enzymes. And all of these, they are chemicals inside our body. So we just want to see the things that are happening inside your body, the reactions, how it is changing to different factors in simple way. Biochemical research can lead to discovery of new therapeutic targets rather, and the development of novel treatments for various diseases. So if you are saying that, okay, we want to see the miracle that happens or the transformation that happens to your normal chemical setting, let me just say it that way, your normal chemical setup inside your body, that's the bio in the biochemistry. Now, we want to now see the changes that happens to it when we decide to introduce something else or when we decide to take out something from it. That's biochemical research. So let's proceed. We are going to talk about some of these important biochemical research techniques. These are not all, but these are like the most common ones. I'm sure we will have gotten to know about some of these already, but let's just have a recap. And for those that do not know them yet, let's just, study together. So number one is chromatography. Chromatography is used to separate and analyze biomolecules based on their properties, usually based on their size. So we have different types of chromatography, right? Based on their size. Let's just focus on that. So what happens is I may not be able to go in depth for each of these, right? But I'm just going to give you a summary and description of each of it. So when we talk about chromatography, we want to, for example, you have a sample and you do not know what it contains, and you have another standard sample and you know what it contains. Talking about biomolecules now. Now, when you subject both to chromatography and separation occurs, when we are talking about chromatography, we will start talking about elution, talking about mobile phase and stationary phase. Now, when both molecules move across the mobile phase, moves across the stationary phase with the help of the mobile phase, you then want to check it on the chromatograph. Chromatograph is like the graph of your chromatography. After you must have run the chromatography, your chromatograph will be plotted for you. Now, what we now get from that chromatograph is, okay, in this particular sample, this is how the graph looks like. Then you remember you have your standard sample and you see how the graph looks like as well. Then you check for which of these standards match or tallies directly or tallies more with this my unknown sample. So that gives you an idea of the possibility of your unknown being that standard that you know before. So it's more or less like, okay, this particular one, if I put it like this, it reaches this height. So this one that I don't, and this one, the name is K, for example. Now this one that I'm bringing that I don't know the name, let me put it next to this. If it's the same height, it means that it's possible that this one that I don't know too might be K. But of course, you know that in research, you do not only do one, you do not only carry out one investigation and draw conclusion. So you do that, and you say, oh, that means there's a possibility that this unknown is this standard, or it will most likely be a species of this standard. Then you can then subject it to further investigations to now confirm if truly it is also okay. That is chromatography, basically. So it works on separation and movement of molecules. Then we have electrophoresis. Electrophoresis is used to separate and analyze biomolecules based on their size and their charge. That's similar to how if you want to apply it, it's also similar to chromatography. You always juxtapose to an already existing standard that you already know its values. Okay. Now, when we talk about spectroscopy, every one of us, if you're a biochemistry student, you most likely use the spectrophotometer. Let me even ask, how many of us have used spectrophotometer before? If you have, just say I have. You have handled the spectrophotometer before. All right, thank you very much, Kennedy. Any other person?
Yes, ma, I
No.
have.
Oh, okay. Thank you very much, Aisha. Thank you very much. So when we're talking about spectroscopy works, it analyzes based on the interaction of light and your biomolecules. Now, let me think of a very simple way to explain this. Okay, when we want to do spectrophotometry or spectroscopy, we have to understand Beer-Lambert's law. And Beer-Lambert's law states that your absorbance is directly proportional to the
I've used it
concentration.
too.
We have used it. Thank you very much. So now, it simply means that when you subject certain biomolecules to light, when we say spectroscopy, the device we use or the machine we use is spectrophotometer. So what we do is when we carry out our assays, so all of these assays, when I'm saying assays, I'll still give you further details. But what I mean is experiments, biochemical experiments. All of these assays have a step, in most cases, the final step, where you be the procedure to be written that you measure the absorbance at a particular wavelength. Now, on your spectrophotometer is a device that works with light. Of course, when you put it on, light travels across it. Now, there is a point where you put in your sample inlet, let me just call it that. So light travels across it and through the sample. So on your spectrophotometer now, you can set a particular wavelength. You can browse more on wavelength and light spectrum and all of that. Now you set a particular wavelength according to your procedure and it measures your absorbance. It gives you your absorbance. Now the absorbance is, okay, how well was your molecule that, or your sample that you have put in, how well is it able to absorb light? Of course, if it is more concentrated, it will absorb, right? If it is less concentrated, light travels faster across it. But basically, what you need to know is spectroscopy rather works on the interaction between light and biomolecules, and it is very easy to understand. Now, let me speak further on it because, like I said, most of us we get to use it. So, when you have your biochemical assays, there are existing procedure for each of your biochemical assays. Now, sometimes, in most cases, this absorbance is always a pointer to a particular biomarker. I will explain the meaning of biomarker. So when they say you should measure your absorbance using your spectrophotometer on a specific wavelength, you just use your spectrophotometer, put in your sample, you set the wavelength, and it brings out your absorbance on the results readout. So you can just take that as your data, as your quantitative data that you need. Now, molecular biology techniques, these ones are not so common, but it's very important. We have PCR, we have DNA sequencing, we have gene editing. So DNA sequencing is in most cases is used, in fact, is used for different things. It's used to study diseases, it's used is used also in genetic engineering. I think some of us would have gotten to know what genetic engineering means, right? But basically, what does it mean? It simply means that you want to know, you take a sample from particular organism, biological organism, of course, a living organism. You take a sample, could be the blood, let's just use blood for example, and you extract the DNA. Of course, when I say extract the DNA, I won't just speak to it and say DNA be extracted. No, it's going to undergo different processes, right? You extract the DNA, and after extracting the DNA, you subject it to sequencing. What you want to do with the sequencing is to understand, um, what's it called now? To understand when we say DNA, we are talking about your nucleic acid, right? And for your nucleic acid, there are nucleotides. So when you say DNA sequencing, you just want to understand how it is arranged. Or sometimes also, when we now link it to PCR, you are trying to relate it then with, in most cases, or an already existing PCR value. Okay, let me break this down. Let's continue from DNA sequencing. So DNA sequencing is, okay, we have extracted the DNA and we want to sequence it. When you want to sequence it, what you just want to know is, you want to know the nucleic acid that it contains in a way. Okay, so when we now talk about PCR is kind of a longer process, but in most cases, what they do with PCR is they use it to amplify DNA. So after you amplification simply means increasing multiplication of an already extracted DNA. So they amplify it. So when they amplify it, then they sequence it. Sequencing it now can either be related to an already existing, I want to see a word that would best describe it. DNA template, for example. So let me say you want to know if this particular person has a particular virus. Now you already have the DNA template of this particular virus that you're working with. Now you want to see, you've gotten sample from this man A, for example, and you want to be sure if he has the virus or to what degree he has the virus. If you subject it to extraction, you get the DNA from the sample, then you subject it to PCR to increase it, to amplify it. Then you now work on it to sequence, and you relate it to your existing templates before. And then with that, you can then see, okay, if it has the same signals like the ones that you've done before or the existing one that you've done before, then it's most likely that this man has this particular virus we are talking about. So it basically works with, when we talk about molecular biology, basically works with DNA, it works with your genes and all of that. Then next thing is enzyme-linked immunosorbent assay. This is ELISA. ELISA is used for measuring biomarkers in the blood or other. Now, let me explain ELISA out of this. ELISA works basically on antibodies, or it works with the antibody and antigen relationship. Like it's here, immunosorbent. So immunology, with your knowledge of immunology, right? Now, antibodies are like proteins in your body, and antigen are like foreign bodies. So when you react it together, or when you introduce an antigen and it reaches an antibody, there is a reaction. So when we are talking about ELISA, ELISA works on that principle. So you are bringing an antigen to an already known antibody. So when there is a reaction, you will know that okay, means that this antigen is present in this sample. Let me break that down further. I take a sample from a particular person, and I want to know if this person has HIV, for example. Now I have my HIV kit for ELISA. Now in that HIV kit, the the reagents in the kit are HIV antibodies. Now I put my HIV antibodies in my plate, and I take this person's sample and I react it with the HIV antibodies. Now I need to mention that even in ELISA, you have to also subject it to spectrophotometer. Now, after including your sample now, and it reacts, if there is a reaction, it simply means that the antigen of that antibody is present in the person's sample. And for the antigen to be present, it means that the person has HIV. So in simple terms, right? That's what it does. So you react a sample with either an antigen or an antibody. And if there is a reaction, you will know that, oh, this sample contains the antigen or the antibody, as the case may be. So very quickly, I want to know if we are really following. Let's have this quick quiz. What are from all of the stories that I've been saying, from all of the examples I've been trying to give, what are the applications and relevance of biochemical research? To save our time, we use biochemical research in various ways. Various ways. Number one is to investigate potentials of certain chemicals, or probably phytochemicals, the therapeutic potentials of phytochemicals. There are quite a number. Another one is for health research, of course. And let me take, for example, my own undergraduate research. What we wanted to do was to investigate the edibility of a particular type of oil that is not common, crepitan seed oil. So it is very affordable and it is cheap, but we wanted to know if we can consume it as humans. Also, we can use biochemical research when we want to prevent diseases. Of course, I've talked about therapy. We also use it for genetic engineering. We also apply biochemical research even in agriculture, talking about genetic engineering and all of that, improving your crop yield, etc. Let's also even talk about nutritional biochemistry. You use your biochemical research to investigate how nutrients affect your health and your wellness. Like, let's remember what I said from the start of this webinar. We want to see how that when you introduce an external factor, you want to see how it alters the chemical reactions within your body. So we have quite a number. We can check more on that. We can do a basic research on that. I already explained to us what basic research means, right? So we can just do a basic research on that. So now, as a biochemical researcher, there is this word that you need to be familiar with, and that is biomarkers. Biomarkers. What does biomarkers mean? Biomarkers are measurable indicators of a biological process or a disease. So I've been mentioning assays. In biochemical assays, or should I say biochemical experiments, your lab work, your assays are used to actually analyze biomolecules. That's what assay means. A process of analyzing biomolecules, enzymatic reactions, etc. So now, biomarkers in itself are measurable indicators of a biological process or a disease. In biochemical assays, biomarkers are used to detect, diagnose, or monitor diseases, as well as to understand the underlying biological mechanisms. Now, I need to explain this to you because when you have an idea of this, when you have an understanding of what biomarkers is, it helps you to know how to navigate your research. It helps you to know in what direction your research will be going. So when I say biomarkers, it simply means your indicators, the indicators that point to a particular thing. So it's more or less like variables that point to a dependent variable. What do I mean? Simple examples that we are familiar with. An increase in your blood sugar is a pointer to the possibility of diabetes. So in that scenario, your blood sugar becomes your biomarker. It means the biological or biomolecular factor that can be used as an indicator for a particular condition. So that's what biomarkers mean. And you get to work with different biomarkers as a biochemistry student during even your undergraduate thesis. Now let's check other examples. So number one here says glucose, like I said, but now let's talk about glucose. Glucose is a biomolecule, right? So someone presents you or your lecturer tells you that, okay, I'm working on diabetes. What do you think we need to measure? The first thing, even if you don't know any other one, the first thing you should know is glucose. Because, of course, when there is an increase in glucose, there's a possibility of diabetes. So when I say biomarker, like I don't know if you heard when I said, but when we are talking about a biomarker, it is simply something that you measure as a pointer to something else. Now, I say if you are given a sample and you are asked to test it for possibility of diabetes, the first thing that should come to mind is, oh, what is a biomolecule that relates to diabetes, right? And that is glucose. So we know that, okay, our biomarker, our measurable factor is glucose. Then we have other examples: cholesterol, we have tumor markers, we have liver enzymes, we have inflammatory biomarkers. For cholesterol is a biomarker for cardiovascular disease. An increase in cholesterol is a pointer to cardiovascular disease. So I'm giving a sample and my supervisor says test it for CBD. I want to know if this person has CBD. I can't just say, oh, do you have CBD? There is no one test for CBD, right? So there are always numerous factors. But one thing that would stand out and what must always be involved is cholesterol. So because it's already established that an increase in cholesterol is equal to a possibility of CVD, a greater possibility of CVD. So I'm measuring cholesterol to know the possibility of a particular condition existing, which is CVD. So we have two more markers too. We have CA125, that is your cancer antigen 125. That one is used to measure ovarian cancer. We have PSA, it's called prostate specific antigen. We have CEA, this is carcinoembryonic antigen. Now, when you measure all of these antigens, they are biomarkers. When you measure them, it tells you the presence or absence of that cancer. Because now, research is continuous. There have been previous research that shows that when there is an increase in this particular antigen, there have been previous years research that shows that in people that have been tested positive for prostate cancer, for example, we saw that there was an increase in PSA activity. So that's already an established research result. It's already an established finding. So now that you have your own sample, you want to know, oh, that means if I check it and there is PSA, it simply means that this person has prostate cancer. Because it has been established that if there is PSA, it means that there is prostate cancer. So you measuring PSA becomes your biomarker. Then we go on to liver enzymes, ALT, AST. We have inflammatory biomarkers as well. We have CRP. This is C-reactive protein. Then this is interleukin 6. You can browse more to understand them. But these are enzymes, biomolecules that point to an already existing disease or that points to a particular condition. So now that now that you have a basic understanding of biomarkers, whatever is your title, whatever is the topic that you want to work with, when your supervisor or when you as a person or as a biochemical researcher, when you know that, oh, these are the rats, I've sacrificed my rats, I've gotten their blood. In this blood, I want to see the person. Probably you fed your rats with, let me see, a very sweet biscuit that is, yes, let's say, what's this biscuit now? Let me use the one that is coming to my head now. Munchkins. Munchkins is a common biscuit now. Munchkins is very sweet. So you want to test if it has possibility of causing diabetes. Now you feed your rats munchkins, you feed them twice a day for 10 days, you feed them munchkins, and after feeding them, you sacrifice them, then you obtain their blood. Now you want to know, there is no one size fits all test for diabetes. But yes, there's something that you can measure that will tell you if there's diabetes. That's something that you can measure as your biomarker, and that is your glucose. So you now measure the glucose level in the blood. If it is higher than normal, you know that, oh, that means this person most likely has diabetes, because there are already existing standards that when it is below this particular number, this particular value, the person is diabetic. So from there, you can of course apply research. You can then say that, oh, we can't eat munchkins daily, or if you want to eat it, you can just eat it once a week. You understand? That is you applying your research. But I think we get the point now, right? All right. So let's go straight into essential skills for biochemical researchers. So the first thing is your laboratory skills. I've talked about some of these techniques, some very important techniques that you must know how to work with or work around as a biochemical researcher. Your ELISA, PCR, DNA sequencing, spectrophotometry, chromatography, etc. Also, pipetting, basic skills, even laboratory practice, safety in the laboratory, they are also important. Then we have analytical skills. Now, as a biochemistry student, I am both pleading and advising that you learn analytical skills. It is very important. Even if there is no provision for an in-depth study or in-depth teaching on it in your school, please and please, you can outsource the knowledge, because your research does not end in the lab. After getting your values, after getting, for example, glucose, you've measured glucose level in like 20 rats. You have measured yes, you have measured glucose level in 20 rats. You have measured enzymatic activities in the rats. You have measured so many factors, so many biomarkers. Let me use the term. You have measured it. You can't just come up and give us the table. No, we want to see it in graphs. I'm sure you must have come across journals like that you see it represented in graphs and some more aggregated and summarized tables. Now, before they could get that table and the graphs, they subjected the results from the lab to analysis. So that's why you need analytical skills. We have so many softwares. We have SPSS, we have GraphPad, and we have some other ones that are not so known that you can learn from. And you must even know how to work with Excel. That's like the basic, right? Then we have computational skills also. This talks about bioinformatics and programming. This is also me pleading and begging. Try as much as possible to learn this. It gives you an edge as a biochemistry student when you have all of these skills, because they are so much in demand. So other things that you need is the knowledge of biochemical principles. I have a lecturer that would always tell us that if you do not know the answer to this particular, if you cannot predict the biomarker for a particular condition, it means that your theoretical background in biochemistry is poor. So also, if you cannot determine the technique to use for a particular test, it means that your knowledge of biochemical principles is actually poor, and also laboratory technique principles. So we really need biochemical principles. We must know the basic A that will be added to a B to give us a C. It makes our work faster. So you must also know experimental design skills. Experimental design is, how do you know the number of rats that you'll be using? How do you know what you will subject your rats to? How do you know when to give treatment? How do you even know what is treatment? How do you know the poison? How do you know the test? How do you know control and all of that? So people don't just come up and say, oh, we want to test for cholesterol, and let's just buy 3 rats now. No, there's always a design. Okay, we are buying 7. Why are you buying 7? You have to have 2 in the control, 2 in the group A, 2 in group B. Now, those in group A, what are you feeding them with? What is the treatment you are giving them? When are you going to sacrifice? Why are you choosing that time to sacrifice? That is experimental design. Your experimental design gives you a direction, and it's always the beginning of your research work. So also, like I mentioned before, you must be familiar with laboratory safety protocols. It's very important. Now, research writing skills, very, very key. And that's what I'm going to take research writing skills very quickly. Like I'm going to try as much as possible to go by the speed of light, even though I know that's not possible. But I'm going to try to quickly rush over it. Research writing skills, because as you know, even if you try to run away or you try to hide when you are being grouped for lab work, you cannot hide from research writing, because you have to carry your cross, you have to carry your individual cross and come up with your report. So that's what I want to quickly talk about now. What are the key components of your research writing? You have your introduction. Your introduction is to give you a background of what you are talking about. Your introduction gives your reader an overview, and your introduction must be catchy. When I see your introduction, I must read it and want to go further. I want to read the remaining content. So provide a context and background information on the research topic. It also contains your research question. Your research question clearly states your research question clearly tells why you are doing your research, what you are trying to find out, what you are trying to investigate, the knowledge you are trying to fetch. That's your research question. So as a reader, I'm reading your work and I'm saying, why did you do this work? Or what is this work about? When I see your research question, I'm like, okay, this is what you're trying to investigate. So your introduction must contain your research question, and it must also contain the significance or sometimes justification of the research you are carrying out. And of course, if your research is not relevant, it is not a research. So you must either be closing a gap or resolving an existing argument or trying to bring in information that does not even exist at all. But you must be bringing what we don't already have. That is the relevance of research. I needed to have mentioned that your research must be relevant. That's why there's a need for literature review. Now, literature review summarizes the key findings and contributions of previous research. It means that if I'm saying that, what's our biscuit now? Munchkins. If I'm saying that Munchkins has a probability of leading to diabetes, in my literature review, I must be able to present previous works that have been done with other biscuits and how that some other biscuits have also been related to diabetes. I must be able to present it. I must also be able to present that, there have been a work that has been done on Munchkins, but that one investigated its effects on lipidomics, on lipids, and not sugar, and not diabetes. Now, I am now coming to study its effect or possibility of causing diabetes, since that one does not already exist. That is literature review. So literature review brings in all existing works that is relevant to what you are studying, and that you can now, at the end of it, you can now tell us, okay, so now this is the gap I'm trying to close. Or you can tell us if it is a situation whereby there have been an argument. Probably a school of thought has been saying that if you drink canola oil as water every day, it is good for your health. While another one has been saying if you drink canola oil as water, it can cause cardiovascular disease. Now you are coming to now say, okay, now that there is this debate, I'm coming to now investigate further under stricter conditions to check if it is truly for group A or if it is group B. So that's the benefit of literature review. So it discusses the theoretical framework and conceptual models relevant to the research as well. There are sometimes that you need to include existing framework, your existing models that you'll be using in your research. You must put it in your literature review because it's also an existing knowledge. Then we have methodology, and it should follow this order. Your introduction is chapter 1, literature review chapter 2, chapter 3, methodology. It describes your research design. Of course, I mentioned that your research design is the first thing. Methodology is like your method. Everything that you did for that research to be successful is what you include. You explain the sampling strategy, the sample size, describe data collection method, outline the data analysis procedures, and even to the data analysis procedures. So for biochemistry now, they want to see the number of rats you use, they want to see why you chose it, they want to see the procedure for each of your assays, they want to even see the place that they want to see a description of your animal house where you reared the animal for your period of research. They want to see it. Like all of those details must be included. Why is methodology important? It is so that your research can be reproducible. So you must give us details that if someone else decides to investigate or to confirm your work, they don't need to come and ask you what you did. They can pick up your work and follow the methods, and they will replicate it. So that's the importance of your methodology. Every detail is important for your methodology. Then you go to your results. That's your chapter 4. Everything that you found, you present it in your result. And please, I need to mention that your results is not going to contain tables of specific activity or enzyme activity. No, you must have subjected it to data analysis and giving it a good presentation, probably as graphs or a summarized table, that when someone sees the graph, they can easily see, okay, this is the significant difference, does not have significant difference, oh, there's significant difference, and all of that. So they must be able to see it on your results. So you can use tables and figures to support your findings and facilitate understanding. It provides a summary of your result of your work, right? Gives us what you got. Then your chapter 5 is your discussion. After you presented your result, now I need to mention that in your chapter 4, you don't need to write the stories. You don't need to tell us details about the results. You can just present the result and just give a summary of the results. You don't need to tell us. You can just now when I say summary, you can tell us, okay, there was an increase in diabetes for group A compared to control group. There was a degree. Did I say diabetes? Sorry. There was an increase in glucose of group A compared to control, or there was a decrease in glucose of group B compared to control. That's all we want to see in your results part after your graph, just short like that. But when you now come to discussion, is when you now tell us that, okay, from that results that I stated earlier, because there was an increase or there was a significant difference, that points to the fact that there is a possibility that munchkins can cause diabetes. I hope people that are working in munchkins are not on this call, but it is just an example, please. So like I was saying, your results should not contain the relating to and saying, oh, from a already existing study, we can say that since introduction of this particular canola oil caused increase in cholesterol, no, you don't need it in your results. Just tell us the significance and insignificant difference there. But when it comes to discussion, we want to see where you now apply it. This is where you apply your research. This is where the applied research comes in. So you acknowledge also in your discussion, you acknowledge the limitations that you may have encountered. Then you suggest future research directions. So your research must also lead to another research. I need to also mention that. We started by saying that it must be relevant. There must be a research gap you are closing. After doing your own research, you must also point out to how people can pick up your work and say, okay, so if you stop here, I can pick it up from here and continue. So we can also suggest future research directions. You can say, okay, I've investigated the possibility of munchkins causing diabetes, but I suggest that further research should be done on how it can lead to cancer. That is you giving future research directions. Also, all of this must be in your discussion. Then you have your conclusion. Conclusion just gives a summary of everything that you have said from A to Z. Gives a summary. It does not have to be too long so that there won't be repetition. Now, but importantly, that's not the end. Referencing. Your references is a list of sources cited in the research, formatted according to chosen citations. So there are different styles of referencing. We have APA, we have Harvard, we have Ankova, we have different types. You can browse more on it. Different types of references. But I think in Nigeria, a very common one is APA referencing style, and that is the one that starts with the author's names, followed by the year of publication, followed by the title of your journal. When I say journal, it means the source of your research, especially for your literature review aspects. You must cite everything. There's nothing like I know it on my head. No, even if you know it on your head, go and browse and look for a journal that supports it so that you can cite it. When you cite, you mention the name, you're acknowledging that I took it from this person's work. So that's where referencing comes in. After, of course, referencing is also related to in-text citation. Whatever you cite in your work is what you will reference. So I was talking about APA. That's the only thing I'll talk about for this particular webinar. So APA, in your in-text citation for APA, if I write Christiana is a female and she finished from Federal University of Agriculture, Abeokuta, if the journal you got it from, if the name of the author is Christiana Igbe and the title is Investigation of Christiana as a Girl, if you want to cite it, when I say in-text citation is in the body of your work. After writing Christiana is a girl and she graduated from Federal University of Agriculture, you put into brackets Igbe, the last name of the author, comma, the year that it was published, then you close your brackets. That's in-text. Now on the referencing list, you're going to write Igbe, comma, C, which is the first name, then full stop, open brackets, the year it was published, then followed by the title, Investigation of Christiana as a Girl, followed by the journal it was published in. Probably it was published in Journal of Igbe Christiana, you put it there, followed by the volume, probably it's volume 2, you put the volume next, then edition, etc. And sometimes you can also add the DOI. DOI is like an identifier. So that's reference, that's APA referencing just summed up for you. Then we have your appendices. Appendices contains other supplementary materials like extra figures and tables. Sometimes we don't give all the details. Sometimes you might have maybe you prepared another food or another diet for your rats to feed on. You don't include that in your main body. You can include the composition of the feed in your appendices. So it's like other information, right? Okay, so becoming an or co-author of a research article. Now I know most of us are already getting information about, oh, you need to you need to have publications even as a biochemistry student. That's when you'll be more relevant, it gives you an edge and all of that. So I just came up with this summary to help you know what it looks like. So I'm going to make it as direct as possible. I mean, I'm going to make it specific to my audience, right? So yeah, the first thing you must, before you can become an author, you can either conduct an original research or you collaborate with others. But let's follow this pattern. So you conduct your original research. We're going to use your undergraduate thesis now. You conduct your original research. In conducting your undergraduate thesis, I mean your undergraduate research, you must identify a research topic, you design and conduct your experiments, you analyze and interpret your results. We've gone through all of that. Then you can also collaborate with others. That's another option. You can find collaborators, find people that are working on a particular thing and join them. You can contribute any of your skills. You can contribute your money as well. Of course, anything at all, just so that they know that, okay, your input is there, then you can be a co-author. But I'm following this particular one, conducting original research. After conducting all of that, next thing is develop your writing skills. I just gave you a summary of what should be included in your undergraduate research writing, right, in your thesis. So your writing skills is important. You come up with it. You read and analyze your research articles. You practice the writing. You get feedback from senior colleagues. And the next thing is you understand authorship guidelines. Browse journals. Journals is like, let me just say, they are like organizations that you submit your work to, then they will now help you publish it. So when you see journal of this, so they are like platforms where you submit your work to and they will publish it. That's why when you see journal articles, you read and you see submitted. No, they don't say submitted. You see the name of the journal. So it means that they were the one that published it. It was published under that journal. So investigate the journal that your research work can be published with. Of course, you can't do a research work on biochemistry and you now say you want to publish in Journal of Management Studies. No, now it doesn't match up. So investigate the journal that fits your work and read through their guidelines. Read through their guidelines. Then, after you must have edited your work to match their guidelines, you can submit to the journals, wait for their review. Most times you get corrections, they'll send it back to you to correct, you work on the corrections, and they will then help you publish it. If it's already fit for publication, they publish it for you. Then, of course, maintain a strong research profile. I need to give you bonuses on this. To maintain a strong research profile, you can write this down. You can create an account on Google Scholar, create an account on ResearchGate, create an account on ORCID, that's ORCID, create account on SciSpace. These are research platforms. So start from them, create your profile. So when you have your profile, whenever you have a publication, you send it there. That's why when you browse on ResearchGate, you see so many authors. I mean, you see so many authors and you see their previous research articles. So you should also go and create your profile. Go and create it tonight to start with. So very quickly, let's have question and answer. I've been talking and talking. Thank you so much for listening to this point. Let's take questions and answers. Do we have any question? Okay, someone said, can I throw more light on the need for experimental design? So basically, experimental design is just like your timetable. It's not only your timetable, right? But it's just like a chart that gives you a direction to what you want to do. So let me use my own undergraduate research and its design as an example. So like I said, we want to investigate the edibility of uraprepitant seed oil, right? Now we're comparing it with canola oil because canola oil has been established as an oil that is consumable, edible to men, to humans. So we wanted to know if this uraprepitant seed oil can also be consumed. So what we did was to get, oh no, I can't remember specific number of rats, but I think we got 25 rats or so. But most importantly, we had different groups. We had the first one, which was our control group. Our control group were fed with normal diet, normal rat food, no additives, nothing. Then we had the HOR group. HOR group were groups that were fed with uracrepitan oil, refined uracrepitan oil, this oil that we are working on. Then we have HOC group. HOC group were rats that were fed with crude uracrepitan oil. Then we had the canola oil group. The group, that group of rats, they were fed with diets containing canola oil. So that's like that already gives a structure to us. Without even getting the rats from our design, we already know we have 4 groups. Now for each group, let's say we need 7 rats for each group. Then we write it down. 7 rats. Group A, group B. Control 7, HOR 7, HOC 7, CO 7. Next thing is we will also write the we also it's still in our design. We also write how many days we will subject it to acclimatization. When you bring in your rats, how many days will they stay before you start feeding them, before you start treating them? You include it in your design. Then you also include when you are going to sacrifice. But the basic thing is always the subjects that you use, whether humans or anything. Then tell us what you gave them. Either you fed them or you injected them. You tell it to us. You state it there in your design. Your design can be a table. You state it there and you write how many days. You write if it is different concentration. You write how many the different concentrations you fed each group. That's part of your design basically. So when you have that design, it then tells you, okay, even if you forget, or even if you are working as a group and group A, and one person in the group is the one coming to feed the rat, and the person forgets to say, oh, how many meals of this chemical do we even give this group, or which group are we even giving canola oil, for example. They can check the design and they say, okay, we are giving the group labeled CO. Okay, how many meals are we injecting them? 5 mil. All right, 5 mil. So that's what your experimental design is about. I hope I was able to clarify that. And contact me via mail. This is my phone number. You can also reach out via LinkedIn. I'm always available. We'll also share links to join our community, building the research community, so that we can get updates on upcoming webinars like this one and workshops.